Hello friends, and welcome to the lecture series on edible vaccines. And in my first lecture, I discussed about the etymology and definition of vaccines and how vaccines came into existence. Then I discussed about the types of vaccines on the basis of their use and their method of preparation. On the basis of their use, we discussed about prophylactic and therapeutic vaccines. And prophylactic vaccines are those vaccines which are administered to the body to protect it against the infections to which the body has not been exposed. Whereas the therapeutic vaccines were meant to encourage the immune response in the body against a particular disorder that already exists in the body or the body by which body is suffering. On the basis of their method of preparation, we discuss about the entire vaccine, wherein we discuss that entire microorganism, whether it was attenuated or weakened or killed or inactivated, complete microorganism is used to prepare the vaccine. Then we also discussed about the subunit vaccines, wherein the specific part of the microorganism, and that specific part can either be a protein or it can be a toxin or it can be a part of the DNA of that microorganism that can stimulate the immune response in the body. So those specific parts are used to prepare subunit vaccines. Then we also discuss about the recombinant and the conjugate vaccines. So friends, in continuation with the previous lecture, today I'll discuss about the ideal characters of a vaccine, followed by the problems with the vaccines which we are using today. Then I'll discuss the definition of edible vaccines, that what are edible vaccines, how the concept of edible vaccines came into existence, what are the advantages of edible vaccines over the conventional vaccines, and their methods of preparation. So coming on to the characters of an ideal vaccine. Now, whether it is a medicine or a vaccine, the prime requisite for it that it should be safe for the user. Since we are talking about vaccines here, so I'll be using the term vaccine more rather than a medicine. So whatever vaccine we have prepared or whatever vaccine is being utilized by the consumer or by the patients, it should be safe for their body. No matter how effective your vaccine is, if it is not safe, then it is of no use. Then obviously the safety is another prime requisite and important character of a vaccine which we are going to discuss ahead. Then the second character is it should be sufficiently effective. So as I told you that efficacy is another important character for a vaccine. So the vaccine whichever you are preparing, it should be efficacious enough to produce immune response against a particular infection for which it is designed or for which it is prepared. An ideal vaccine should have an easy route of administration. So as I discussed in my previous lecture that most of the vaccines are administered through the parenteral route or with the help of an injection, which is not a patient compliant method of administration. The vaccines, we should develop certain vaccines that have which are more patient compliant, or we can say which can be administered through an easy route of administration. Then the vaccine should have a simple and easy method of preparation or production, because their method of preparation will have a direct impact on the cost of the vaccine. So more complex method, more sophisticated method, higher will be the cost of the vaccine, which is not an ideal character. So the vaccines should have simple and easy method of production. These vaccines should also have ease of handling. When I talk about ease of handling, that is post-production handling of these vaccines. As I discussed in my previous lecture, that majority of the vaccines require refrigerated conditions for their storage as well as their transportation. So this also adds up to the cost of the vaccine, which sometimes make the vaccine costly in comparison to the cost that is being inflicted in their method of preparation. So majority of the cost comes from their handling factors, that is their storage or their transportation. So whatever vaccine we are preparing, it should have an ease of handling, specifically post-production handling of the vaccine should be quite easy. Then last but not the least, vaccine should be cheap. it should be affordable to the person belonging to every strata of the society. Because if the medicine or the vaccine is not cheap, it is costly, it will defeat the purpose, it will defeat the goal of providing the healthcare facility to each and every person of the country. Now we move on to the problems with conventional vaccines. Most of the problems are same as the characters of an ideal vaccines. First one is the safety. So as we discussed that the vaccines which we prepare should be safe, but on the other hand, safety is another issue or the problem with the conventional vaccines that are prepared by attenuation or inactivation of the microorganism. Because it has been observed, as I discussed in my previous lecture, that some of the microorganism or viruses, they do not get fully weakened or attenuated or inactivated, and they can multiply in the human cells, thereby showing their toxic effects. Or they can get converted into certain, they can mutate into certain virulent strain which will exhibit the toxic effect. And this we discussed by taking an example of oral polio vaccine, which has been discontinued in some of the Western countries, because it has been observed that oral polio vaccine or the virus, it gets mutated into a more virulent strain which causes paralytic polio. And therefore, majority of the oral polio vaccine has now been discontinued, and inactivated polio vaccine is now being used in place of the oral polio vaccine. Then the other problem is the need of storage in refrigerated condition, and not only storage but transportation in the refrigerated condition. And as we have discussed in the ideal character, that such kind of handling infrastructure will eventually add up to the cost of the vaccine, which is obviously not desirable. Now, most of the vaccines, as we discussed, are administered by the parenteral route, and it demands need for the trained personnel. And as we discussed, that the parenteral route is not the patient compliant route, and in addition to that, it also requires certain trained personnel to administer the vaccine. Because if you have to consume a tablet or any medicine orally, then obviously you don't require a trained person. You can take the tablet and you can swallow it. But if you have to administer the medicine with the help of injection, you cannot do it by yourself. For that you require a trained personnel. So this is another drawback of the conventional vaccines. Now, in addition to the requirement of the trained personnel, it has also been observed that these vaccines which are administered parenterally, they exhibit certain secondary side effects, such as inflammation at the site of injection, or some people suffer from fever, or some exhibit certain hypersensitivity reactions. Though all these secondary effects vary from person to person, but these have been observed with these conventional vaccines. Then, as we discussed, that some of the vaccines, specifically subunit vaccines, they have lower immune response in comparison to the entire vaccines, and therefore they require multiple doses or the booster doses in order to have a sustained immune response or sustained immunity in the body. Then it has been observed that injectable vaccines stimulate only systemic immune response. Now, what is systemic immune response? For that, I will just give you a brief idea about the type of immunity or the type of immune system we are having in our body. We have broadly two types of immune system. One is innate immune system, which is also known as non-specific immune system or the immune response, and the adaptive immune response or the specific immune response or the immune system. Now, innate immunity or innate immune response is in our body since our birth. So whenever a child is born and is exposed to external environment which is filled with microbes, still that newly born baby the body of that newly born baby protect itself from those microbial exposure. And this is largely because of that innate immune response or the system in our body, which is highly non-specific. It will not recognize that what kind of pathogen it is, it will just kill that pathogen. Whereas in case of adaptive immune response or immune system, we produce antibodies that are specific against a specific microorganism or a microbe. So the injectable vaccines or the vaccines which we are using today, they only stimulate the systemic response, which is largely the adaptive response, because we are injecting an antigen and against that antigen our body produces antibody. Now, apart from this systemic immune response, mucosal immunity is an important aspect of our body. And what is mucosal immunity? How it shows its action in our body? This we will discuss in detail when I will discuss about the mechanism of edible vaccines. Then it has been observed that in case of conventional vaccines, that it is not possible to develop vaccine against all the diseases by the conventional methods. And for certain disorders, we require certain sophisticated or highly costly methods which increases the cost of their production and makes the vaccine unaffordable for a common man. So friends, these were certain problems with the conventional vaccines. And now we move on to the edible vaccines. Now one can say that the concept of edible vaccine it originates from the saying of Hippocrates, "Thy food, thy medicine," means let your food be your medicine. Though this saying mainly highlights the importance of eating balanced diet, as it has been observed that if you eat nutritious or balanced diet since your childhood, or you have a very nutritious dietary pattern, that then you can delay the onset of certain degenerative disorders. But one can easily correlate the concept of edible vaccines with the saying of Hippocrates, "Thy food, thy medicine. Let your food be your medicine." Now it all began in the end of the 20th century when Hyatt and co-workers in 1989 tried to produce plant-based vaccines. But it was Dr. Charles J. Ernsten, a famous plant molecular biologist, who gave the term edible vaccines in 1990. Now he was greatly influenced by a program that was launched in 1990, and that program was named as the Children Vaccine Initiative. The major goal of this initiative was to harness new technologies in the quest for the ultimate vaccine, including being affordable, being heat stable, and orally administered. Because the vaccines were, this initiative was for children vaccines, so the major goal was that it should be easily administered, it should be affordable and heat stable. So this kind of goals stimulate the thought process of Dr. Charles, and he started working in this field of the preparing edible vaccines. And therefore he is considered as a pioneer in the development of plant-based vaccines for human disease prevention, with special emphasis on the need of poor countries, because as we discussed that some of the poor countries don't have infrastructure for the storage and transportation of the vaccines, which require refrigerated conditions. So this was the major stimulation behind this kind of work. So later this concept was used to develop effective therapeutics against the biowarfare or bioterrorism. So Dr. Charles also developed vaccines against Ebola virus using tobacco plant, and he also developed transgenic plants for Norwalk virus and hepatitis B. And during his study, he found that whenever the animals were fed on those transgenic plants wherein the antigen has been introduced in the plant, their body developed antibodies against those viruses. Or in simple words, the body of the animals became immune against those viruses. So he defined edible vaccines as transgenic plant and animal-based production of or those contain agents that trigger an animal immune response. So in simple words, we can say that edible vaccine, since we are dealing with mainly with the plant-based vaccine, so I will say that in simple words, edible vaccines are the transgenic plants which contains specific antigens. So whenever those plants are consumed, those antigens enter into the body of the user or the consumer, and those antigens initiate the immune response against a particular infection for whose antigens are being administered. Now, why edible vaccines are required? Now, the major reason behind is the problems that we discussed under the conventional vaccines. Edible vaccines are considered as safe because we don't require needles to inject the vaccines, and when we don't require needles, there are less chances of infection. Then these have easy route of administration. That is, they can be administered orally. And as we have already discussed, that oral route is the most patient compliant route of administration. Then since these vaccines are administered orally, no specialized trained personnel are required to administer these vaccines. These edible vaccines have cheaper production cost in comparison to the conventional vaccines, and therefore they are more cheaper. They also have ease of handling post their production. That is, they don't require any kind of refrigerated storage conditions or transportation conditions. In simple words, they are stable toward higher temperatures. When we say higher temperatures, it means room temperature or slightly heat conditions. And these edible vaccines, they provide both mucosal and systemic immunity. They stimulate both mucosal and systemic immunity, and as we discussed in case of conventional vaccines, which only stimulate the systemic immunity. Now we move on to their methods of production. Now their method of production can be broadly categorized on the methods which are used to introduce the desired genes into the plant genome. Because as we discussed, edible vaccines are nothing but these are the transgenic plants. So in these transgenic plants, we introduce the desired gene or the antigenic material into the plant genome. So on those basis, we can classify the methods of preparation of edible vaccines into indirect gene delivery methods or the vaccines that are prepared by the direct gene delivery method. Now, whenever the vaccines are produced by the direct gene delivery method, they require certain carrier or the vector to transfer the gene into the plant material. And therefore, these are also known as vector-based methods. And these include utilization of agrobacterium to transfer the gene into the plant genome, or simple words, agrobacterium-mediated production of the edible vaccines, or genetically engineered plant virus-based production of the edible vaccines. Whereas the direct gene delivery method includes the ballistic or the gene gun method to produce edible vaccine. Now, apart from these, there are several other methods to produce transgenic plants, but we will be mainly focusing on these methods. The other methods are using polyethylene glycol and using electric current, though I will be discussing the use of electric current in transferring the gene that is largely known as electroporation method. I will discuss that method briefly ahead. So coming on to the first indirect method, that is agrobacterium induced method. Now agrobacterium is a gram-negative bacteria that attacks the plant and transfer their genes to the plant nucleus. Mainly two species of agrobacterium are used. One is agrobacterium tumefaciens and second one is agrobacterium rhizogenes. Now agrobacterium tumefaciens contains TI plasmid, which is nothing but the tumor inducing plasmid. And this plasmid is responsible for causing the crown gall or the tumor gall disease in the plant. Whereas agrobacterium rhizogene contain RI plasmid, and this RI plasmid is responsible for causing hairy root disease in the plant. Now before we move on that how this agrobacterium is used to transfer the gene, first I will just give you an brief overview that what is the structure of TI plasmid. Now as you can see in the picture that TI plasmid contains two major portions. One is known as tDNA, which includes the genes that induce tumor. Further, it also includes the genes that are responsible for increasing the secretion of auxins and cytokinins, which also assist in the induction of the tumor in the plant. Then this tDNA or the tumor DNA is surrounded by two corners or the two flanks. That is left corner or the left flank and the right corner or the right flank, and both these flanks have a 25 base pair repeat. Now the second portion of this TI plasmid is the virulence gene portion, which is also known as Vir portion or the Vir gene portion. And this Vir gene portion is further composed of 7 different genes which are known as Vir genes. And these include Vir A, Vir B, C, D, E, F and Vir G genes. Now how this bacteria induces tumor in the plant? What happens is whenever the plant is wounded, it has been observed that whenever the plant is wounded, it secretes certain phenolic compounds, specifically ectosyringon. Now these phenolic compounds act as the major attractants for the agrobacterium tumefaciens, because I will mainly focusing on the agrobacterium tumefaciens because this is the most widely used bacterial species for producing transgenic plant. So these phenolic compounds, they basically attract these agrobacterium tumefaciens, and they bind, these phenolic compounds bind to Vir-A receptors that are present on the cell wall of the bacteria. So as these phenolic compounds bind to Vir-A receptors, they cause phosphorylation of these receptors. This phosphorylation further stimulates the Vir-G proteins. Now these Vir-G proteins further stimulate different Vir genes that are present on the virulence gene portion of the TI plasmid. First in the sequence is the Vir-D1 and D2, which are nothing but endonucleases, which cleave the desired part of the tDNA at the 25 base pair repeat portions, that is left and the right flank of the tDNA. And this cleavage produces a single stranded DNA which is attached to a molecule of Vir-D protein at one end, mainly 5 prime. Now when this Vir-D is attached, in the meantime Vir-E proteins also bind to this complex of DNA fragment, single stranded DNA fragment along with Vir-D. Now this binding of Vir-E protein protected from the degradation of nucleases. And in the meantime, Vir-B genes also get activated, and these Vir-B genes, they form a pore-like passage between the bacterial cell wall and the plant cell wall. And this pore helps in the transferring of that tDNA complex, which includes tDNA, Vir-D and Vir-E molecule into the plant cell. And once this complex enters into the plant cell, this tDNA gets incorporated into the plant genome, thereby inducing the tumor formation in the plant. So this is how basically this bacteria infects the plant. Now how plant molecular biologists or biotechnologists use this mechanism to produce transgenic plant? What they do is they remove the gene from the tDNA that is responsible for the induction of tumor, right? Once this gene is being removed, the gene of the desired character is inserted in its place, and the gene of desired character is the antigenic gene that will induce the immune response in the body. So that particular gene is inserted in the tDNA molecule, and the tumor inducing gene is removed. Now this TI plasmid is again inserted in the bacteria, and the plant is infected with that particular bacteria. Now since the tumor inducing gene has been removed from the plasmid, the plant will not get infected from the tumor, but the plant genome will get incorporated, or the desired gene that has been introduced in the TI plasmid will get incorporated into the plant genome. Now what happens is the antigen or the gene which is responsible for inducing immune response is incorporated into the plant genome, and since it is present in the plant genome, it will be present in different parts of the plant which are to be consumed. So once the person will consume that plant part or the fruit of that plant, that antigen will enter into his body and will stimulate the immune response. So this is how agrobacterium is used to produce the edible vaccine. The second indirect method is the genetically modified plant virus. Now in this, epitope of the antigen is identified. Now epitope is that part of the antigen or the microbe which is responsible for inducing the immune response. And then the DNA fragment encoding this epitope is fused with the gene of a plant virus like tobacco mosaic virus, thereby producing a recombinant virus. Now sometimes what happens is entire gene is fused with the entire genome of the virus, thereby producing a chimeric virus. Now chimeric virus is nothing but it's a virus that is produced by the fusion of the two desired genes, in which one can be responsible for the replication and other can produce the immune response in the body. And these two desired genes, they are obtained from the two different microbes or two different viruses. Now such modified viruses are used to infect the plant, thereby producing the modified genes into the plant genome. And once this desired virus enters into the plant genome, there is a rapid replication of this viral genome, and numerous copies of the gene are produced inside the plant, thereby producing higher level of expression of those genes, which are the antigenic genes. But the major limitations of this method are that viral reproduction product must first be purified from the infected plant before they are vaccinated. So during the replication of the virus inside the plant, certain byproducts are formed, and these byproducts should be first removed. It means the viral antigen should be first purified. Then these plant viruses by which we are infecting the plant, they will cause the plant death following the infection. And therefore, when the vaccine is caught, another plant needs to be infected with the virus, and this reinfection method needs to be continuously made to produce the sustained vaccines. Now we move on to the direct method, and the most common direct or the most widely used direct method is the ballistic gene gun method, which is also known as microprojectile bombardment method. Now, since no carrier is used in this method, it is also known as non-vector gene transfer method, and this is largely used when vector methods are not possible or are not successful in certain plants. Now, in this, desired antigen DNA or RNA is coated with gold or tungsten microparticles, and then they are loaded into a gene gun. Now, particles are then bombarded onto the specific plant part with the help of high pressure that is generated by the helium gas. Now, these bombarded particles will enter into the plant system, and then the desired gene gets incorporated into the plant genome. Now, by this method, we can produce two kinds of transformation. One is the nuclear transformation, wherein the desired gene is incorporated into the nuclear genome of the plant, and second one is the chloroplast transformation, wherein the desired gene is incorporated into the genome of the chloroplast. Now, amongst these two, chloroplast transformations are found better for various reasons, but the main reason of our interest is that the chloroplasts have high ploidy, and therefore they show better expression of the foreign protein, leading to better accumulation of the desired protein. And further, multiple genes can be expressed simultaneously by using chloroplast method, which is not possible in case of nuclear transformation. Electroporation method, and in this, targeted cells and the desired DNA are kept together in a suitable solution, buffer solution, in the electroporation cuvette. A suitable strength of electric impulses are passed for microseconds for specified period of time, and these impulses causes reorientation or change in the structure of the cell membranes, thereby creating pore inside those membranes. Now, these creation of the pore allows the desired DNA to enter into the cell and get incorporated into the genome. Now, these pores that are created by this electroporation method are temporary, and gradually the pores again seal themselves. Now, the major advantages of electroporation method is that this method is fast, it is less costly in comparison to the other methods, and we attain stable transformations by using the electroporation method, and large number of cells can be treated by this method. So friends, this was all about the methods of preparation of edible vaccines, and in the next lecture I'll discuss about the mechanism of edible vaccines, that how these vaccines produce immunity in our body, and vis-a-vis we will also discuss what are the various plants that are being used to prepare or to create or to produce these edible vaccines. Thank you.